overview of today's presentation, the outline. I'm going to give a brief overview of the PrEPVacc trial. So this has already been presented by Eugene Rezagia a while ago as part of the International AIDS Society virtual workshops. So I'm just going to give a brief overview of it and give a mini status update on where we are in regards to the trial. Then I'll give a detailed overview of a somewhat detailed overview of the PrEPVacc statistical analysis plan. And from presenting the analysis plan, we're going to look at some novel aspects of the PrEPVacc trial and their relevance to the future design of HIV prevention trials. So some of these aspects we're going to discuss will include how we're using the multi-arm, multi-stage design, how we're using a hypothetical / counterfactual placebo arm in some of the PrEPVacc analysis, using them with the averted infections ratio, using that hypothetical counterfactual placebo arm with the averted infections ratio. We're also going to discuss some of the how relevant the registration cohort is to how relevant having a registration cohort like the PrEPVacc trial has, how relevant it is to the future design of HIV prevention trials. And then we're going to look at some of the strengths and anticipated challenges with the methods we're using and the current proposed solutions for those challenges. So the PrEPVacc trial is one, I think we'll just have to use this view. So the PrEPVacc trial, the main primary study objectives include assessing the safety and efficacy of 2 HIV prophylactic vaccine regimens, comparing each regimen to placebo. We're going to assess how effective they are in reducing HIV incidence. And then the second trial objective is to compare the safety and efficacy of Descovy to Truvada in reducing HIV incidence. I believe most of us are familiar with Descovy and Truvada. These regimens have been shown to have some level of effectiveness and efficiency in preventing HIV. And for Truvada especially, there is a lot of data on it in various populations, heterosexual men, heterosexual women, MSM, and the like. But then with Descovy, there is a bit of limited information on its effectiveness among heterosexual women. So the PrEPVacc trial is aiming to contribute to more information on that. An overview of the treatment arms for the PrEPVacc trial. We're going to have participants who will be enrolled at week 0, and they'll be randomized to one of 3 treatment arms. That's DNA, HIV, PT123, and AIDS vax administered at weeks 0, 4, 24, and 48. Then you're going to have the second active vaccine regimen of DNA, HIV, PT123, and TN54 administered at weeks 0 and 4, and then they get then a follow-up of MVS, CMDR, and TN54 administered at weeks 24 and 48. And these 2 vaccine arms will be compared to a placebo arm where the vaccines will be administered at weeks 0, 4, 24, and 48. So across all our 3 trial arms, you have the vaccines at 0, 4, 24, and 48. And then in addition to the vaccines, there will be a concurrent 1-to-1 randomization comparing 2 PrEP regimens. The PrEP component of the study will be open label. So at baseline, the participants will be randomized, yes, to the vaccines, but they will also be randomized to receive Truvada and Descovy. And study PrEP will be dispensed to the participants all through until 2 weeks after that immunization. That will be around week 26, after which PrEP will revert to locally registered supply of the generic drug. So within the first 26 weeks before the vaccines are estimated to kick in, we'll be able to compare the efficacy, the effectiveness of the 2 PrEP regimens to the other. We'll be assessing whether Descovy is as good as Truvada. Then from week 26 onwards until week 74 and beyond, we're going to be comparing each of the active vaccine arms to a placebo arm and see whether the vaccine arms essentially are better than placebo. So the PrEPVacc communities, we have Fisher Fall populations and other populations in Masaka, Uganda. We have bar workers and female sex workers in Dar es Salaam, Tanzania. We intend to include female bar workers in Mbeya, Tanzania, a general population sort of group in Durban, South Africa, and key populations in Maputo, Mozambique. So those are the planned groups to include in the PrEPVacc trial. However, in preparation of the trial, there is an ongoing registration cohort. So our registration cohort has been ongoing at all the 5 study sites, and it has been recruiting participants and following them up. Currently, over 2,500 participants have been enrolled and followed up in a registration cohort at the 5 study sites. The plan is that it's from this registration cohort that we will enroll participants into the trial. And this registration cohort will continue alongside the trial, even as the trial progresses, but with the plan that whoever is in the cohort is eligible to be enrolled into the trial. Current status update before we get into the statistics. The PrEPVacc trial has been activated currently at 1 study site, that's in Masaka, Uganda. We intend to start at the other sites in the next few months. There is an ongoing registration cohort, as we mentioned, at all the 5 study sites. We currently have about 2,551 person years of observation in the registration cohort and have observed an incidence of about 3.2 per 100 person years. COVID lockdowns did affect attendance of visits in the registration cohort a bit, but we have been mostly able to attend at least about 71% of the visits in the registration cohort. The trial just started and we have very few participants enrolled, and we haven't yet missed any visits as yet. So that's in a nutshell where we are. Now let's get to the statistical aspects of the PrEPVacc trial. So the PrEPVacc trial, as we saw from the upper slides, it has 2 main objectives. One is to assess vaccine efficacy. The other objective is to assess PrEP effectiveness. Now, vaccine efficacy. So the PrEPVacc trial is powered to detect a vaccine. If a vaccine has an efficacy of 70%, the PrEPVacc trial is planned to have 90% power to detect this vaccine efficacy. So we estimated that 500 participants followed up in each trial arm who potentially could accrue 20 events. For example, in the placebo arm, it would be enough to provide this 90% power to detect that 70% vaccine efficacy if these participants are followed up for the planned 48 weeks after the 26th week. So you have to be in the trial for the 74 weeks, but we estimate for the weeks in the vaccine analysis, that's the number of participants we need. So in total, the trial is planning to recruit 1,668 participants. With 3 arms, we'd have about 500 in each arm and leave enough room for about 10% loss to follow up. So that's the vaccine efficacy sample size aspect. Then for the PrEP effectiveness, we estimated that the 834 participants, if you're to split our entire trial population into the 2 arms, the 834 participants followed up for those first 26 weeks to provide reasonable power, that's about 90% power, to show that at least if there is 50% preservation of effect provided, 50%, there'll be 50, we'll be able to show that if Descovy has at least about 50%, showing that at least 50% preservation of effect, provided that Truvada effectiveness is at least 70%. So if the Truvada is at 70%, we have enough power to show that there is at least 50% preservation of effect by the Descovy arm. And then our overall assumed placebo incidence rate for the trial is 4 per 100 person years. The registration cohort has shown about 3.2. We expect to recruit even higher risk participants from the cohort to power the trial at 4 per 100 person years. The overall trial design is a phase 2B, 3 and 2-stage trial. It's an endpoint-driven multi-arm, multi-stage trial. The trial timelines, just a simple overview. We have the vaccinations at weeks 0, 4, 24, and 48, and we're able to follow up these participants to week 74 and beyond. The PrEP analysis will cover mostly from week 0 up to week 26, after which we're going to have the vaccine analysis from week 26 and beyond. There'll also be routine HIV tests in the follow-up period beyond week 74. So our participant is able to stay in the trial up to week 74 and beyond. So the analysis for the primary vaccine efficacy. Our primary outcome will be HIV acquisition. We're going to first focus on the vaccine efficacy, then the PrEP effectiveness. So now just for the vaccine efficacy, HIV acquisition by a participant who completed their first 3 immunizations and was HIV negative at week 26. So if after the first 26 weeks, you are known to be HIV negative, then we follow you till the end of the trial. We assess whether when you get HIV infection, or we follow you up until you're censored from the study. The analysis for this will be a time to event analysis using a Cox proportional hazards regression, which is adjusted for site and sex. Vaccine efficacy will be 1 minus the hazard ratio for each active arm. We're going to report this with 95% confidence intervals and p-values. For the vaccine efficacy bit, the handling of missing data. So this one being a time to event analysis, participants will be followed up until they're censored, until their last HIV test. So any missing data after that will be considered censored. But if in case we're missing the key week 26 result, it will be handled depending on the scenario. So for example, if someone misses the week 26 result, but is HIV negative on a follow-up assessment, they'll be considered to have been negative at week 26. So this is a bit key in distinguishing the two analyses, but I don't need to go too much into detail. But if they're proven to be positive beyond week 26, even though they missed their week 26, we'd consider them on their first positive result in the vaccine analysis bit of the study. So there'll be some, so I won't go into detail on that. So there'll be some supplementary analysis. We intend to follow, there'll be a supplementary analysis following up the participants strictly from week 26 until up to week 74 to strictly show us a participant followed up for 1 year after vaccination, what's the vaccine efficacy. And then we're also going to have a supplementary analysis, which will include all the data from week 0, including all the participants from week 0 with all their data, and we will assess, follow-up begins at week 0 instead of 26, and we assess them to see when they seroconvert and if there's a difference in the two arms. So there would include all the participants who received any vaccination. Then the other aspect is the safety and secondary vaccine outcomes. I'm not going to go too much into this slide, but our main primary safety outcome will be a clinical decision to discontinue the vaccine regimen for an AE report related to the product. So we'll compare that across the two study arms with proportions, rates, and 95% confidence intervals. So that's it really for the vaccine analysis. At least that's what I've prepared for this presentation. Now for the PrEP effectiveness, we will assess the HIV acquisition by a participant before week 26. They're followed up, they were negative at enrollment, they're followed up until week 26, and we see, did they get HIV or did they not? The participants will be randomized to Truvada or to Descovy, and we'll compare the number and rate of infections presented for each PrEP arm. We'll compute rate differences and rate ratios across the two different PrEP arms with 95% confidence intervals. Participants with a missing 26 result will be censored at week 26. And we're mainly going to use the averted infections ratio for this analysis. I'm going to get into the details on that on the next slide. And we intend to also have some supplementary analysis assessing PrEP efficacy with possibly using a bit of case analysis or considering a certain threshold of adherence. So there will be a supplementary PrEP efficacy analysis. So the averted infections ratio, I hope by now most of us are a bit familiar with this ratio, but the averted infections ratio in a nutshell, it's the proportion of infections, in this case, that should be averted by using Descovy relative to using Truvada. So if we're to realize, it's almost similar to a normal incidence rate ratio, except for its addition of the rate in the placebo arm. And if the two arms are similar, the averted infections ratio would be 1. If the Descovy arm is a weaker arm and has a higher HIV incidence, the averted infections ratio would be less than 1, and so on and so forth. The key thing to this ratio is it's using a rate in a placebo arm, and these are PrEP analysis, so obviously we don't have a placebo arm. However, the way we are estimating the incidence in this placebo arm, we're combining 3 different approaches. For one, we're using the incidence in the observational pre-trial registration cohort. Now, this bit, it's not as easy. One, we intend to use this alongside with using possibly propensity score matching and ensuring that the population we're comparing from the registration cohort is as similar as possible to this trial population. But of course, we can't really be, it's not a perfect science. We wouldn't have created an actual placebo arm, but it gives us an estimate. The second option is we'd use the incidence in the, so one key thing is people behave differently once they're in a clinical trial than they would in a registration cohort. So that's kind of the gap there. One of the other options for estimating this placebo incidence is we use the incidence in the vaccine placebo arm after week 26. Remember, we have 3 vaccine arms, and then one of the arms is actually a placebo arm. So we use the rate in that placebo arm to give us an estimate of what the underlying placebo incidence may be. Once again, it's still not perfect, but it gives us an idea of what this incidence could be. The third approach is we know, okay, there is information on the estimated effectiveness of Truvada. We will be collecting adherence data in this trial. So if we know how effective Truvada is, we know how much the participants are adhering, we may be able to estimate a background placebo incidence. Once again, it's still not perfect, but it would give us an idea. So the fourth option is we combine, we do a synthesis of all these 3 approaches and try and reach an estimate of this hypothetical placebo. So I picked this graphic from some of the Discover trial analysis. So just to inform this discussion. So as part of the PrEPVacc trial, we'll conduct sensitivity analysis, robust sensitivity analysis, examining how altering the assumed placebo incidence rate impacts this averted infections ratio and its confidence limits. In particular, we want to see, we know that lowering the hypothetical incidence rate will produce averted infections ratios further and further away from 1. And as you're doing that, that actually is less favorable to an inferior drug in that equation. The inferior drug, it should be making it go further and further away from 1. And so the idea would be at what incidence rate are you able to show this drug is clearly inferior? And then you ask yourself, is this a plausible incidence rate? That's that sort of idea, but the whole idea is we're going to have robust sensitivity analysis informing this background placebo incidence rate. And this slide is on the safety and the secondary PrEP outcomes. I will not go too much into it, but our main primary safety outcome will be a clinical decision to discontinue the PrEP regimens for an AE considered related to the product. Yeah, and then descriptive analysis of the other secondary outcomes. So routinely, we will have independent data monitoring committee reviews of the data. There will be the biannual IDMC reviews of the accrued safety and incidence data. So this will be by treatment arm and by treatment arm. So we're going to review both the data for the vaccine and the PrEP, not we, the IDMC. We'll review both the data for the vaccine and the PrEP as it's being accrued to assess its safety and the incidence data. There is one planned formal interim analysis to assess vaccine efficacy after we approximately have 7 HIV infections in the placebo arm. At that point, when we have the 7 infections in the placebo arm, the IDMC will look at the number of participants with infections in one or both of the arms. Now, if the number of infections in the placebo arm is the same as the number of infections in an active arm, the IDMC could potentially recommend stopping further enrollment to that arm after reviewing additional analysis such as conditional power and confidence intervals and the like. But at that point, they would at least be able to rule out 70% vaccine efficacy, and it then becomes a question of, do they stop the trial or let the arms proceed? So some of the aspects of the PrEPVacc trial that are relevant to the future design of HIV prevention trials, one is the MUMS design, which broadly speaking, it's a novel paradigm for conducting adaptive trials, which allows several treatments to be assessed concurrently with pre-planned interim adaptations. And it's been said to have sizable gains in efficiency over conducting a series of in comparison to a single stage 2-armed trial. It's a lot more efficient. And of course, this depends on the availability of these other treatments, but the HIV prevention field is changing rapidly, and we would hope for a future where we would be able to have several different alternatives, in that in the middle of a trial, if you're able to rule out an intervention not working, you're able to replace it with another one that needs to be tested and that sort of thing. So the MUMS design is an efficient approach which could potentially be used in future HIV prevention trials if we keep, if new prevention interventions keep being identified. The other novel aspect of the PrEPVacc trial, which is relevant to the future design of HIV prevention trials, is the use of the hypothetical counterfactual placebo arm. So most future trials will not include a placebo or a treatment arm. So the PrEPVacc PrEP analysis and approach, they offer insight on what this could look like, the use of a robust sensitivity analysis and the like, and the different methods of estimating the placebo incidence. And then also we're using this registration cohort running before the trial and also continuing into as the trial progresses. And it provides useful contextual information. It provides information on how the available interventions in the community to prevent HIV are being used. It gives you a nice estimate, a reasonable estimate of the placebo incidence rate, and is actually quite useful to the overall analysis of the trial. So some of the strengths to our statistical methods and designs of the trial is, as mentioned, the MUMS design, which is efficient, and it is a method that can actually keep up with the quickly evolving landscape of HIV prevention interventions. The use of the averted infections ratio, which requires significantly smaller sample sizes than the traditional incidence rate ratios. Now, this will be more useful, especially with the development of highly effective HIV interventions. Some of the challenges, of course, are still about being able to accurately estimate this background HIV incidence. And our solution to this for now is combining several approaches of estimating the background incidence, with the registration cohort data being quite helpful, and using robust sensitivity analysis around this. And I'm coming to the end of the presentation, and I'd like to acknowledge the several statisticians who have contributed to this statistical analysis plan. They are the blinded statisticians, that's Christian Holm-Hansen and me at MRC UVRI. Christian Holm-Hansen is the senior trial statistician. Then you have the independent statisticians at MRC CTU who have contributed as well to the statistical analysis plan. That's David Dunn, Angela Crook, and Henry Van, the unblinded trial statistician. I think he may be on this call. And then there are, of course, the independent data monitoring committee statistician, Doug Taylor and Christopher Ware, and other members of the IDMC who have also reviewed the SAP and contributed to some of its elements. And on that note, I'd like to thank you all so much for listening, and of course acknowledge all our funders and collaborators in this trial. Thank you.